I like that there are some themes that are kind of running from one talk to the next, and I think that's going to happen again, perhaps. Our fourth speaker is Lisa M. Coussens, professor and chair of the Department of Cell, Developmental, and Cancer Biology, and deputy director of the Knight Cancer Institute at Oregon Health and Science University. The U.S. government has spent more money to fight cancer than it has to address any other disease. Tremendous progress has been made since 1971, when Mary Lasker convinced President Richard Nixon to declare war on cancer. Yet cancer remains high on the list of unmet medical needs. Dr. Coussens, who recently served as president of the American Association of Cancer Research, has made cancer her life's work. It's clear now that cancer is not one disease, it's many diseases, and cancerous cells themselves are only part of the story. Their success, the cell's success in creating and advancing tumors, depends on a complex ecosystem, including a dynamic tumor microenvironment with blood vessels, supporting cells, and other normal cells that can influence tumor growth and resilience. In addition, a cancer patient's immune system can play both cancer-promoting and cancer-suppressing roles. Dr. Coussens' group works to understand this complexity with a goal of determining how immune regulation and inflammation can be intentionally manipulated to treat cancer. Her group has developed technology to study the immune context of cancer, to understand its human biology, and to move treatments forward in the clinic. Elected to Class 4's section on medical genetics, hematology, and oncology, the title of her talk is Inflammation and Cancer, From Basic Mechanisms to Therapeutic Targets. Dr. Coussens.
Thank you so much. It is a tremendous honor to stand in front of you today and deliver this lecture that really is not possible or would not be possible without many that came before me and the talented young minds that I have had the extreme privilege of mentoring in my career. But I'll start with a little bit of history. I'm the youngest of three in a family. The youngest of over 30 first cousins, the youngest in my entire generation. So I held a special place in my family. When I was little, a young girl, I was asked, what did I want to be when I grew up? Very intelligently, I said I wanted to be a helicopter. Not the pilot, the machine. My older siblings nearly peed themselves when I said this, and continued throughout my lifetime to remind me what a little geek I was. When I defended my PhD, those were the first warming words my brother said to me, still a geek. But I rapidly grew out of that phase and turned to creativity and thinking that I wanted to be an artist. Was awarded several awards in high school based upon out-of-the-box designs to think about how to express what was in my mind. Talent is required to be an artist. In college, I rapidly realized an income I was never going to earn. And so I turned to my next greatest love, biology. Having grown up outdoors with my parents and my siblings, the natural diversity within the palette of biology in our backyards was really what consumed me. My undergraduate degree was in marine biology and ecological sciences, where I was just in awe of the beauty of Mother Nature and schlepping around tide pools at all times of the day and night. Little did I know how appreciation of that diversity would serve me really for the rest of my career. Because I was an aquatic biology student, in essence, my very first job professionally as a scientist was taking care of a Xenopus laevis colony at Genentech in the early 1980s. Now, to put this in historical perspective, this was long before there were companies that sold kits for everything that we do today. And so I learned how to take care of the frogs, harvest the eggs, and inject them with some of the early clones when Genentech was looking for some of the early interferon genes. But fortunately, companies like NEB and Biorad rapidly emerged, and I was put out of business. Now, one might think, oh, that's career flaw or fatal career blow because no more job. But what happened was perhaps the greatest strike of luck in my life, in that my position as a technician was acquired by a man that you may know, Axel Ulrich, who won the Lasker Prize several years ago for his discoveries and help with developing the drug Herceptin. One of my very first cloning projects was EGF receptor, but also the HER2 oncogene. Little did I know at the time the degree to which that fundamental and seminal contribution would change the way of breast cancer mortality and morbidity, with that women who are diagnosed with HER2 positive disease have a much longer and improved outcome because of the drug Herceptin. But what that did was it infused in me a passion for science. Bitten by the bug, it was clear to me that for the rest of my career, it was going to be spent in the context of cancer biology. When I did my PhD, I was very frustrated with mere in vitro technology and thinking about gene expression and the impact of altered expression of oncogenes in cells growing on plastic. I knew I wanted to study cancer in vivo in appropriate context. Well, at the time, transgenic models of multi-stage cancer development were really just becoming available for use. Doug Hanahan's work with expressing oncogenes behind tissue-specific promoters gave us the very best window into the natural history of cancer development and gave us the first opportunity to evaluate pre-malignant biology in addition to a malignant tumor, which is where most of cancer biology had been prior to that. What struck me with being a postdoc in the Hanahan lab was the progression that we were able to see and the reality of that mere oncogene expression wasn't sufficient to develop a fully malignant and/or metastatic tumor. What was clear was that the surrounding microenvironment and the systemic response to that oncogene expression and that malignancy was co-opted by the oncogenic signaling in order to facilitate tumor growth. Being a biologist in training, what made sense to me was that a reductionist approach would be able to identify what critical components outside of the tumor cell were really important for manifesting that tumor. And what captivated me the most was, having really been a marine biologist, I knew nothing about tissue pathology or the progression of a tumor. And so I took it upon myself to learn pathology and conducted several electron microscopy studies of normal tissue, pre-malignant tissue, and fully malignant cancer. And what images captivated me is one on the top here that shows one invading malignant epithelial cell migrating through a basement membrane. Now, what I had learned as an undergraduate and early in my career was that cancer was a tremendously chaotic and disorganized process. But what that one image showed me was how regulated the process of invasion was. And what a very wise mentor pointed out to me in that image, Zena Werb, was that if you look just to the left of that invading epithelial cell, you'll see a macrophage holding the gate open in the basement membrane, which told me there was tremendous communication and collaboration in the invasive process. I didn't really know about myeloid cells, macrophages, or the immune system at all at that point. And so I asked the fundamental question, how many of these immune cells are there in that pre-malignant tissue? In the lower panel, what you'll see is immunohistochemistry staining for a cell that we just heard about called a macrophage. And what I found was that there were just as many macrophages present as there were supposed pre-malignant tumor cells, and they're shown in red, telling me something was very important about that initial immune response, and very much akin to what Rudi Virchow had proposed over 100 years prior, that cancer arises at places in the body or in tissues that are suffering from chronic inflammation. And so as I started my own lab, we started out looking at what is the state of chronic inflammation across common solid tumors. Is that inflammation the same in context, or instead, is it sculpted by the local microenvironment? Thinking immunologically, the pathogens that are present in the lung are quite different than the pathogens or assaults that a mammary tree in the breast or the prostate or anywhere else in the body would be exposed to. Because after all, the immune system isn't there to ward off cancer. The immune system is there to fight infection and to maintain tissue homeostasis. And so as we conducted significant studies, what we found was that in chronic inflammatory diseases, there's a very significant increased relative risk of malignancy in those tissues, telling us that that inflammatory assault was very, very important. As we collaborated with surgeons, we wanted to understand the context of those inflammatory assaults. And as I said, what I'm showing you here in brown is called CD45 staining. CD45 is a protein expressed by all cells of the immune system. And so what you can see in these very normal homeostatic tissues of the breast, the pancreas, the lung, and the pleura of the thoracic cavity is that there are pre-stationed resident immune cells. In the lower panels, what we've done is collected those tissues and subjected them to flow cytometry, where we've used antibodies enabling us to quantify cells of distinct immune cell lineages. You don't need to know which ones they are, but suffice it to say each color represents a distinct immune cell subtype. What you'll see across these four homeostatic tissues is that the composition of the immune cells present are quite distinct. Again, that makes sense because those tissues aren't subjected to the same kinds of pathogenic assault. So the question then becomes, in a malignancy that has chronic inflammation, is that inflammation all the same? Or again, is that sculpted by the local microenvironment? You can see on the top, looking again at CD45, that inflammation is chronic in all of these malignant tissues. But if you utilize the same approach as shown by flow cytometry down below, what you'll see is those chronic inflammatory infiltrates are indeed distinct by tissue, but more common to the resident microenvironment that's present. That was a very important observation because what it told us is that if we could block this inflammation or quell the chronic inflammatory signaling, we had to think in a tissue-specific way about the regulatory mechanisms driving this process. We also have to think about the spatial aspects of these immune cells. Many years later, we now have technologies where we can spatially audit where immune cells are in tissue. And what you can see here on the bottom, red are malignant epithelial cells, and in various colors, you can see that there's different architectures and spatial patterns of those immune cells where we have major categories of how the immune system is organized in solid tumors. So we now have to think not only about the contexture, but the spatial organization of these immune cells with regards to the kind of communication networks that they're involved in. And so with these fundamental images in my mind, where the lab has come from with our research questions has been to try to understand these diverse ecosystems and understand how those diverse ecosystems drive these tumor cell extrinsic biologies that are just as important as the oncogenes or tumor suppressor genes and mutational, the genes subject to mutations that are driving aberrant proliferation and subsequent tumor development. But to fundamentally ask the question, where are there vulnerabilities in those regulatory pathways that we could leverage therapeutically to quell the process? One of our very first or early indications that we might have luck in identifying these vulnerabilities came from work from David DiNardo when he was a postdoc in my laboratory, where we identified the CD4 T cell, just as you heard previously, as being a very important T cell, not for killing tumor cells, but for educating the macrophage through cytokines that we just heard about called IL-4 and IL-13. As it turns out, T cells are recruited into early pre-malignant lesions, quite likely as a fundamental wound healing response, but they're very rapidly co-opted by that neoplastic or malignant microenvironment to stimulate other immune cells that are co-opted to turn on programs like angiogenesis, lymphatic angiogenesis, fibrosis, and other fundamental aspects of what we see within the hallmarks of cancer. And what David recognized was that the macrophage was very important for regulating tumor development and metastasis, but if we took out the CD4 T cells or blocked the communication from the T cell to the macrophage, tumors grew more slowly, and metastasis in these animals never happened, telling us there would potentially be a therapeutic target there to block pre-malignant progression, but also potentially to block end-stage metastasis. Well, what is a macrophage and where does it come from? The signals, all tissues have macrophages pre-stationed in them. And a macrophage is one of the very first immune cells that responds to damage, any kind of damage. Paper cut, stub your toe, surgery, an infection with a virus or bacteria. Macrophages are there to try to fix the problem and get that tissue back to a homeostatic state. Where they come from is from embryonic tissue or from hematopoietic stem cell progenitors. And they arrive in tissues as a function of a cytokine or polypeptide growth factor called colony-stimulating factor 1. Now, brilliant work in the early 1980s and early 1990s from Jeff Pollard and Richard Stanley's lab had identified a mutant mouse that didn't have macrophages in any of its tissues. And through early gene mapping studies, they demonstrated that that macrophage deficiency was actually due to a dominant mutation in the CSF1 gene. They subsequently went on to show that in the absence of CSF1, there were no macrophages in mammary cancers in animals, and the absence of CSF1 led to delayed tumor development and diminished metastasis, very much like what we had seen with the CD4 T cells regulating the macrophages in our breast cancer models. And so we hypothesized that that represented a sufficient therapeutic vulnerability where we could target the CSF1 signaling pathway and shut it down and change the natural history of inflammation in those solid tumors. And so we've worked with several drugs that target the CSF1 receptor signaling axis. CSF1 receptor is a receptor tyrosine kinase whose kinase activity can be turned off by small molecule inhibitors that block the ATP binding activity within the receptor kinase. Antibodies to either the ligand, CSF1, or to the receptor can block ligand binding and block stimulation of that receptor. We set up several models. The most notable that we utilized were mouse models of mammary cancer development, also developed in the early 1980s. But we chose one that had an abundance of macrophages within its mammary tumors. Shown here is flow cytometry data again, looking at major leukocyte lineages within those mammary tumors. And what you can see in these tumors is upwards of 80% of the cells within a tumor represent the macrophages. The drugs, if we give the drugs to animals with malignant tumors, we can see a progressive decline in the presence of the macrophage. If we take animals off drug, you can see that those macrophages rapidly come back in because they're recruited in from the bone marrow and other hematopoietic stem cell sources. And so we evaluated the degree to which these could slow tumor development or potentially block metastasis. And what we find is shown here on the left in a tumor growth kinetic assay where we're prospectively monitoring tumor growth. Black is the control animals, no treatment. In light blue is just monotherapy with the CSF1 antagonist. In orange is standard of care chemotherapy that women with breast cancer get in a neoadjuvant setting. But in red is the combination of the CSF1 receptor antagonist with chemotherapy. And what you can see is a very significant slowing of primary tumor growth. But more importantly, if you look at the lungs of these animals, where tumors naturally, with 100% penetrance, metastasize to the lung on top, the big dark blue staining, but on the bottom you see a complete absence of those, saying that the drug plus chemotherapy had blocked that late-stage metastatic progression. What was notable about this study was the fact that when we shut down that macrophage pathway, we saw more CD8 cytotoxic T cells that were active against the tumor that set up the biology for tumor cell killing. And that was what was responsible for the decreased cell growth. If we take out the CD8 T cells, what you can see here in light blue is you completely lose the benefit of that combination therapy. This was the first study to show the dynamic communication network between an innate immune cell, the myeloid cell, and activation and functional significance of a CD8 T cell within a solid tumor. Now, we've asked many questions with regards to this paradigm, and what we have found that across tumor types, as shown here in black, the macrophage represents a pro-tumorigenic immune cell that drives survival pathways, inflammatory programming, angiogenesis, hypoxia, and very importantly, T cell suppression. If we block these pathways and block the programming of the macrophage, what we find is functionally relevant pathways where those macrophages are now known to inhibit T cells. So whereas the sculpting of how macrophages become pro-tumorigenic is tissue-specific, the biology downstream of that macrophage as a paradigm is conserved. They're pro-tumorigenic and T-cell suppressive. And when we identify these critical leverage points and treat with drugs that block them, we find commonality where we reverse the transcriptional programming of these cells and turn those tissues back more into a wound-healing microenvironment where tumor growth is slowed and end-stage tumor development is greatly diminished. Now, we've been fortunate to collaborate with medical oncologists and take several of these approaches into the clinic where we've translated our work with drugs, some of which are now FDA approved, where we've, in a neoadjuvant or in an adjuvant setting, conducted proof of principle clinical trials that what we're seeing in the mouse is conserved with the biology that we see in women with disease or pancreatic cancer or people with recurrent head and neck tumors. We've gone on to identify that this macrophage T-cell ratio is conserved across all tumors, whereas a signal for more aggressive monitoring or more aggressive therapy, if you stratify patients by a macrophage T-cell ratio, you can identify the patients that will have a worse outcome with regards to progression-free survival or overall survival, telling us that we can use contexture of the immune system to identify therapeutic approaches for patients in the tumor types that we've evaluated, but really across the board with some of the worst malignancies that people suffer from. And so the questions going forward are to understand how these diverse macrophage-mediated tumor-promoting pathways can be quelled to enhance clinical outcomes for patients with tumors. And because I ran long, I will go through my last 4 slides very, very quickly and get to the punchline, where these mouse modeling experiments and the proof of principle clinical trials have told us that for immune therapies, we have to listen to the biology, and we have to think about the tissue-specific mechanisms that are driving those immune responses in order to shut them down, and recognize that Mother Nature and the tumor is always going to win, and so we have to be prepared to monitor response and resistance mechanisms to go forward with additional therapeutic tools. And I want to leave you with hope, because we know that since the war on cancer was started, we're doing much better. And we're doing better because of fundamental investments in basic science and technology, some of which have been driven by the people that are in the room and people that are members of the National Academy. With that, thank you to the people over the last 25 years that have been in my lab. I am deeply embedded to them. This honor for me is really a testament to their passion, their intelligence, and their insight. And thank you to the funding agencies that have thought and funded out-of-the-box thinking, and all of my collaborators through the years. Thank you.
I think we could take one question. She was obviously trying to get out of it.
I was.
Very important work. All right, she did get out of it. Good. Thank you for your talk.